Thank you for coming back and carrying on with my videos in my tilapia series. Today's video is part 3 on nutrition. Now, tilapia nutrition has been started back in the 1980s and up through today. There are thousands of papers dealing with the various aspects about tilapia nutrition published by researchers in all parts of the globe. So it's very difficult for me to talk about nutrition in any great amount of time. And so I'm really just skimming over some of the key points that I think are most important in understanding some of the real core ideas that I share with tilapia nutrition. So without further ado, let me get started. So in this video, it's around 45 slides or so. It'll probably take about an hour to do. So sit back as I share my information with you. The first thing I'm going to talk about are nutrient requirements. And if you haven't done so, please go and look in the Nutrient Requirements playlist here in YouTube and watch the first video on theory and reality of nutrient requirements, and then the second one on trials and studies, so you get a good idea about how nutrient requirement studies are conducted and why we have to use them as guidelines rather than as absolute values that we just simply use in our feed formulation. So I've said it before, and I will say it again, there are many factors that affect nutrient requirements. And this is just a short list. So we have characteristics about the fish itself, its physiology, age, growth stage, sexual maturity, and energy intake. And then we have external factors in the environment. So we have water temperature, water salinity, stress, and oxygen intake, or in other words, dissolved oxygen in the water. And then we have other factors external, such as pathogens, parasites, potential infections and diseases, and maybe we have dietary limitations. Uh, Furthermore, we often will have interactions with other nutrients. So for example, we have one limiting amino acid and it impacts the utilization of all other amino acids. So let's first look at protein and amino acids. Now, I have said in my other videos that a requirement for protein does not really exist, although it's a very simple way to understand it. Actually, what a protein requirement is an estimation of the amount of protein required to provide an animal with the correct amount of amino acids, both essential and non-essential, that it needs in any given day to grow, assuming that we know something about its physiology and we know something about its feed intake. So here we have from this book, Tilapia Culture by El Sayed, uh, a list of protein requirements of cultured tilapia. And we can see that we have fry, fingerlings, gift tilapia adults, we have gift males, and we have quite a number of other species. So we've got Mozambique, Aureus, Zilii, and then we have some hybrids down at the bottom. But if we go over to the requirement column, which is this column over here, what we see is that even though we're talking about, say, um, Nile tilapia, it's interesting that the protein requirements that are published are all over the place. And so that should kind of give us an idea that maybe there isn't an absolute perfect requirement that we should use, but we have to understand that each one of these researchers has used a particular kind of fish, a strain of fish. They've used their own feeds. They have their own R&D facilities. They have their pluses and maybe some deficiencies in their research protocols. And maybe they have done a good job with their statistical analysis, and maybe they haven't. And so then these numbers all come out. So we just have to look at them in totality and try to decide which ones we want to follow rather than just picking one and saying this is it. So I'm not going to focus a lot on requirements, but I'm going to talk about some of the things that are relating back to that first table that I showed you, things that impact the amount of protein, or in other words, the amount of estimation of the amount of amino acids that a fish might need. But here we're going to look at some of the factors that, let's say, modulate that idea about a protein requirement. So the first one here is digestive protein modulates digestive enzyme activities and gene expression in red tilapia juveniles. And there's the QR code. You can go and download that paper for free. So what I'm showing you here is we have the basic uh, information about the diets. I'm not showing you the diets themselves. You can go and read that in the paper. But we see we have 24% protein, 30%, 36, and 42. And then we see the crude energy, the amount of fat, ether extract, the ash, and the total carbohydrates. And then we see in the table underneath the different characteristics relating to the growth performance. So we have initial weight, final weight, specific growth rate, SGR, and then we have something about the whole body composition of the tilapia after the end of the experiment. And when we look at what they were actually testing, which was the digestive enzyme activities, so if we look at one of these things, we see we have protein content across the bottom, 24, 30, 36, and 42. And that applies to all 6 of these graphs. And then we have acid protease. So acid protease is basically just lumping a bunch of proteases together. These would be the serine proteases, trypsin, chymotrypsin, but they're shown here individually. And then we have pepsinogen expression. Now, when we talk about pepsinogen, or we're talking about an enzyme with a gen on the end, it means this is a proenzyme. And it is produced, but because they don't want it to work inside the cells and cause damage wherever it may be, they're sent in an inactive form, but when they reach the tissue where they're supposed to be active, like the liver or the lumen of the gut, then there are other enzymes that remove this and turn it into the active form. So pepsinogen produces pepsin. So pepsin is something that we typically will find in the stomach, where all of these other enzymes the trypsin, the chymotrypsin, um, are found in the uh, posterior, primarily in the posterior intestine. And then as the intestine begins to raise the pH to become alkaline after the pancreas, then we would move more into alkaline proteases rather than acid proteases. But what I wanted to point out with this slide was if you notice here, 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 that as we get to 36% protein, we see for here and here and here, and basically also here and here, that the highest expression of the proenzymes and the expression of all of these proteases occurs at 36% protein. And then it decreases at 42% protein. And I think the way best to understand this is that somewhere between 36 and 42, because we don't have details between these points, the ability of the animal to digest and absorb protein or amino acids, digest the protein and produce amino acids for absorption, it becomes saturated. And the animal basically says, well, I don't really need any more protein to digest because I've got all I need at the moment. And so then it begins to down-regulate the amount of acids and enzymes that it is secreting. And so that's why we see this drop. So somewhere between 36 and 42, we have reached really the saturation point for this fish to digest the proteins into amino acids. This paper is called Interactions of Varying Dietary Protein and Lipid Levels in Young Red Tilapia. So, and then it says evidence of protein sparing. So what this implies when we talk about protein sparing, it means that for generation of energy, fish can use protein for energy, fats for energy, or they can use carbohydrates for energy. And what we do not want to do is we do not want to have the fish burn protein, catabolize protein as an energy source. We would rather that protein be used for making muscle tissue, for making meat. So we want to find a way to have the fish use energy either from fats or from uh, carbohydrates. And if we do that successfully, then protein will be catabolized less or maybe not at all, and it will be used for building muscle instead. So their research implies that when we get the fat level in the feed and the protein level in the right balance, that we actually can spare protein for growth and reduce its use as a source for energy. So in our first graphic here on the left-hand side, average daily growth compared to protein intake, we see along the bottom here that we have protein consumption in milligrams per gram fish body weight per day. And so we have 40, 60, 80, 100, 120, and 140. And then we have average daily gain in percent on the vertical axis. So if we look here, we can see that we have 15% protein, 20% protein, and 30% protein. And so that's our little codes we can use for white circles, black circles, and white triangles. And then we also have 4 different fat levels. So we have 6% fat, 12% fat, 18, and 24. And if we look at the line that we have graphed here, we can tell by the color and by the code which protein level it is and which fat level it is. And if we look here, we see that the highest average daily growth is coming from diets with 30% protein and 10 and 12% fat. And all of the triangles are more or less here at the top. Then in the middle here, the black dots representing 20% protein, and then all right down here at the bottom, we see protein 15%. So this tells us, looking at the vertical axis, that actually 15% protein did not grow very well at all. 20% grew faster in terms of daily weight gain, and 30% seems to be better. However, when we go and look at the graphic over here, so we see ADG, FCR, PER, protein efficiency ratio, and NPU, or net protein utilization. And we notice here that for the 30% protein diet, we have the highest daily gain. And somewhat lower is the 20% protein diet. However, when we go and look at PER, or protein efficiency ratio, so this is the amount of feed fed in relation to the amount of protein deposited. So we know how much protein is in our feed, and we do this calculation. What we see here, although this was the highest in terms of gain, it was the lowest of the three in terms of protein efficiency. So this makes me wonder up here that this is not actually weight gain coming from muscle deposition from protein. It's coming from deposition of body fat, because it's very easy to add fat and the fish will gain weight relatively quickly, but it's not very useful for us. Likewise, if we go over here and we look at the FCR, we see that the lowest FCR, which is a good thing, is with the high protein diet at the higher fat levels. But once again, I believe that this is due to rapid deposition of fat, and it's not due to protein deposition. And we see here that 20% protein is just kind of more or less level, indicating that the feed intake and feed utilization is quite more or less consistent. Then when we go down and we look at net protein utilization, which is a measure of protein in versus protein deposited, if you want to know what how that is calculated, go back and look at my nutrients video on protein and amino acids, and I explain how these numbers are calculated. So here we see that we have a relatively high net protein utilization, and the two lower protein diets are more or less the same at that point. Okay, well, that gives you an idea that they suggest that we have a better balance, maybe 20% protein at not such a high fat level. We actually get better protein deposition, which in terms of what a fish buyer wants, they want to eat meat. They don't want to eat fat. And so that seems to be a better combination than going for a high protein diet. Now, just keep in mind that on that previous table, we were showing you that these protein requirement levels were all over the place. And so from this page, we then begin to understand that we have to realize how much fat, how much dietary energy those diets that they tested had, and what was the source of the energy, whether it was coming from carbohydrate, from fats, or coming from protein. Okay, so here's another paper. This one is quite recent, and from 2022, interactive effects of protein and energy intake on nutrient partitioning and growth in Nile tilapia. So we had all male tilapia, so monosex tilapia, 63 grams weight. We had 8 levels of digestible protein intake, and you can see in this graph up here what the intake was on a daily basis: 1.9 grams, 2.1, up to 6.16. And then we have also two levels of non-protein digestible energy intake. So they very carefully made their diets so that they had a protein diet that was all based on very purified protein. So we have casein, we have soy protein concentrate, we have pea protein concentrate, and some fish meal, about 25%, 22% of each of those. And they did not really provide any fat to speak of. Only the fish meal had some fat, and none of the others has any fat. And then also very little carbohydrates in any of those. And so then they had able to provide carbohydrates from energy, from starches, and also from fats. And then they have premix that they used in common. So when we look at protein intake, and we see that in the top graph here, everything is just, superimposed one on top of the other. But if we look down at the bottom, it seems to show to us that the energy intake naturally is going to be higher. The black circles are naturally going to be higher because they're getting 22 kilojoules per day, and compared with the low energy, which are getting only 18 per day. So that's why the black line is, consistent across all proteins, but the uh, low energy diets have a lower energy intake. So same paper. Now, if we look at this one, we see here we have our digestible uh, uh, pro protein and energy intake. Here we have body mass gain. So that's basically weight gain in grams per day. That's equivalent to an ADG. And what we see is as we increase protein levels, then the growth rate increases. And it seems that the animals benefit a little bit more from the high energy intake than the low energy intake, because the black dots are slightly above the white circles. But when we look at body fat content, we can see that the high energy diet is leading to actually quite high fat levels in the body. And in some cases, we look up there, that's basically double what we see down here. And what does it show us? Well, it shows us that having low protein and high fat is a bad idea. That's going to lead to an overly fatty fish. But as we begin to get into the right level where we have some kind of a balance between uh, protein intake and energy intake, and they're balanced, we see that we have everything more or less merging together down in this area. Now we go over here and we look at protein gain. Well, the dietary protein content is the same for both of those. And here we're basically not considering the fact that weight gain includes fat as well as protein. So here we're just looking at protein gain. And so this makes sense if we don't count the fat, that the weight gain should be very similar based on protein intake. But when we go to the next one, fat gain, we can see that definitely there is more gain in body fat with the high energy fat, or the high energy intake. And that there's some relationship that as protein levels go up, there's a slight increase in body fat deposition, which kind of makes sense because then more protein is being deposited and the fat is not being used as a not being that useful. It's just being deposited instead. Now, when we look down here at protein efficiency, so this is PER, we see that as protein increases and then we see the PER is going down. Now, that's similar to the last couple of slides that I've shown you, showing that as protein levels in the feeds increase, actually the necessity of fish to absorb them most efficiently goes down. The more they have, the less they have to work at extracting every last amino acid out. And so we saw in that slide where we went from 36% protein, the energy or the enzyme levels were high. And when we went to 42, the enzyme levels dropped down. This is the same thing that you're seeing right here, that the protein efficiency, the animal is just simply not converting all of the protein into amino acids and absorbing it. Okay, so this is another paper. Actually, I like this paper quite a lot. And so here we have a paper from 2020, feeding graded levels of protein to genetically improved farm tilapia. So those are gift tilapia and reared in inland saline water, effect on growth and gene expression. And so then we have these growth factor genes. And so we have gift tilapia. They're very small, only 2.68 grams. And they were being fed a purified casein gelatin dextrin diet. And then they were reared in 10 parts per thousand salinity. And the reason for using a purified diet, casein gelatin for protein and dextrin for energy, is just simply to remove a lot of the confounding factors. So what do we see here? We see we have weight gain compared to crude protein levels. So look at our dietary protein levels. We have 15, 20, 25, 35, 45, 50, and up to 55% protein. And so they've plotted some linear regression lines here, but then they've also done this quadratic curve. And what we can see is that based on the lines that they get a value right here, 37.86. So they're saying based on the broken line or the straight lines that 37.86% protein is better. However, when they're using the linear or the quadratic equation curve here, they find that the best level, and I mentioned in one of my other videos, we typically will pick 95% rather than 100%. So we're not at the maximum of, or the apex of the curve, we're slightly below it. And they're saying that the protein level is a little under 41% at the maximum there. So if we look at this graph on the right-hand side, we're seeing dietary protein requirement compared to liver IGF. So IGF is a growth factor. It's a gene, and expression is compared to crude protein level. So same protein levels, and then we have the polynomial equation giving us the curved line there, and then we have the broken line, the linear, giving us those X-shaped lines. And in this case, in terms of IGF-1, the gene expression is more or less the same for both protein levels. Now that last one was done at 10 ppt salinity. Here, what they've done is they've compared gift tilapia 3.25 grams reared at 0 ppt, so fresh water, and at 8 ppt salinity. So this paper, Culture Salinity Alters Dietary Protein Requirement, Whole Body Composition, and Nutrients Metabolism, related genes expression in juvenile gift tilapia. And this is a paper from 2021. I've given you the year in red, meaning that this is not a paper that you can download for free. But if you have your institutional or your university um, subscription through your library or whatever, then you should be able to get access to this because it's in a well-known journal. Okay, so let's look here at the graphic on the left-hand side. So we have dietary protein levels. So we have 10, 20, 30, 40, 50, and 60. And then we have feed conversion ratio. So we see that we obtain with the uh, zero salinity is this line right here, and 8% salinity is the lower line here. We see that at zero salinity that the optimal protein level seems to be about 33.7. But when we have 8% or 8 ppt salinity, that the protein level that gives us the lowest FCR has the inflection point at around 37.7. Okay, so we see this is slightly lower, but that protein level is excessive, and it just doesn't make any sense to have a protein level that high for basically nothing gained over the inflection point right here. Now, if we go and look at weight gain, so this is expressed as percent, this might be equivalent to a, I guess it's just percent weight gain against our protein level, and we have here then the 8% salinity is the upper line with the black dots, and the 0 ppt salinity is the lower one. So what this tells us then is that the tilapia seem to be able to grow slightly faster at 8 ppt salinity than they were able to grow at 0. And that the optimum protein was between about 37.9 and 38.2, so quite a narrow range right there. Very close together, where over here we saw that there was a spread of about 4% in terms of the suggested requirement. So this is interesting because then it suggests that actually these gift tilapia actually can grow better in let's say brackish water rather than in true fresh water. Okay, so that was everything about protein that I wanted to talk to you about. And so now we just have some tables. Here we have the amino acid requirements. This is really why we want to be eating the protein, so we can get the amino acid that we require. And we see here for Nile tilapia, we've got Mozambique tilapia. We have for essential amino acids, arginine, lysine, methionine, cysteine, phenylalanine, leucine, and isoleucine. And you can see that the research has been conducted from as far back as 1988 up to 2009. Carrying on with the remainder of the essential amino acids, threonine, valine, histidine, and tryptophan, and you see them. When we look at the parameter, all of these were done using only weight gain. And I do not particularly like weight gain because it's very susceptible to changes in uh, rearing conditions, and there's a lot of factors that can have an implementation, have an impact on weight gain. And what I do like, though, is that all of these requirements are expressed as a percent of crude protein. So that doesn't matter whether it's a 25%, 28, 30, 32, 34, 36. When it's expressed as a percent of crude protein, we just simply take our protein level, and 3.8 or 4% is going to be our requirement for threonine and for histidine. Now, as we see salinity affecting protein levels, it's probably not surprising that salinity also will affect the utilization of amino acids. So here we have this paper from 2023, Salinity Levels Affect the Lysine Nutrient Requirements and Nutrient Metabolism of Juvenile Gift Tilapia. And so we have tilapia 6.24 grams, reared at 0 and 8 ppt. And then we have dietary lysine supplementation. So 1.34, 1.7, 2.03, 2.41, 2.72, and 3.04% of feed dry matter. And here we have low salinity is the black triangles, and we have high salinity as the black diamonds. So if we look at the inflection points here, and what these people have done is that they've chosen to just simply give us the apex of the quadratic equation, and then they have just gone down. So here we see that the amount of lysine here is 2.13%, and over here is 2.31%. So rather than just saying one or the other, we then could say, well, maybe it could be somewhere in the middle, because this is, you know, not much. It's about a change of about 0.2%, which is not really very much. But we just understand that once again, that we see that it's slightly higher requirement at the higher salinity. Now, when we go and look at feed conversion ratio, so this is, feed eaten versus weight gained on a wet basis, we see actually somewhat greater difference. So we see 2.15 compared with 2.13 for the low salinity. And then for the high salinity, we see that it's actually a little bit higher at 2.45. So that's a difference of about 3.34% difference. Once again, I guess the key point I want with these couple of graphs that I've shown you, this slide and previously, is that we have to take into account the salinity of the water where we're growing the fish, and that may then require us to make some adjustments in both protein level in the feed, but then also how much essential amino acids we are providing in that protein. Okay, moving on to lipids and fatty acids. Once again, this table from the El Sayed book, and we see here Tilapia zilii, Nile tilapia, blue tilapia, hybrids of various kinds. We see gift tilapia and several experiments of the gift tilapia and so on. And then we see levels tested, percent of diet. So, for example, the very first one there, we see levels tested were 1.7 to 15% of diet. So this is fat. Now, when I see requirement as percent of diet is at 15, it means that they found the best result at the highest fat level that they used. And going back and thinking about how nutrient requirements are obtained and some of the common mistakes that people make is when you have a requirement by simply saying that was my highest tested level and that was the best, you should actually redo that experiment. And to say for them that 15 was the best, how do they know that it wasn't 17 or 18 or 20? Because they didn't have levels up there. So it's not actually a very good idea to simply test and say your maximum level was the best. It means you made a mistake. You should have tested beyond that point. If it was me, I would redo the experiment. Anyway, once it's done and you've packed everything up, you can't go back and do it again. Now, if we go and look at the second one here, we see that they also say 15, so that's interesting. But notice that they did 3 to 19, so they did some levels above 15, and then they were able to find maybe a maxima at lower than the maximum fat level that they tested. Third one, same problem. They said 0 to 10, and 10 is the best. No, I don't really accept that. Then we see this one with Mozambique tilapia. They tested 6 to 24. They said 18 is the best. And so once again, that maybe is a more reliable thing. You go back and look up the research here. This is from Dr. De Silva. I mentioned him earlier. And you can go and see how they did the experiment themselves. So one thing you can see is we have quite a wide range of recommended dietary levels for fat, all the way from 3.6 up to 18. And does it really make a lot of sense that it would be so varied like that? And so then we really need to go back and look at each one of these studies and see what was the diet that they used. Because the diet that they used may have had an impact on their determined fat level. And it's the research protocol that's given us all of these variations. I don't really think that the fish in reality have a requirement spreading across this far. I would expect probably somewhere in the vicinity of 10 to 15, 12 to 15 is probably a better result. And we see the top several results are more or less in that range. So we look at the tilapia fatty acid requirements, and we have really only two that we're concerned about, and that is linoleic acid, 18,2 and 6, and then we have here N3 fatty acids. So what that's really referring to is linolenic, 18,3 and 3. And in all cases, we see here that there's a requirement for linoleic 18,2 and 6, uh, and there's a requirement, but here in 1999, which is quite a long time ago now, it was required, maybe by doing some kind of a qualitative test, but the quantity had not yet been determined. And this is coming from the National Research Council Nutrient Requirements of Fish and Shrimp in 2011. And it's a bit of a surprise that for hybrid red tilapia that nothing had been published later than 1999 to be included in their book, because they could have taken research up as late as, say, 2009, maybe even 2010, before they released the final version of this book. Okay, vitamins and minerals. So we look first at fat-soluble vitamin requirements. So, we know them, right? Vitamin A, D, E, and K, and we have hybrid red tilapia, Nile tilapia, blue tilapia, and we have requirement, and in many cases you can see we have weight gain, but we also have several other things like maximum liver storage and several other factors. I do not remember what the ADS and the AASLP refer to, so you could go and look at those papers. Those were published in NRC 2011. And then we have in vitamin E, female tilapia. This was published by Nacimiento in 2014, so it was not in NRC 2011. And they were looking specifically at breeding performance. And they state that 400 for maximum reproductive performance, where just for growth and for health, we see that all of the others were more or less in the range of, say, 50 to 100. And in the case of vitamins A and D, normally they are listed in terms of international units, where um, vitamin E is normally recommended in milligrams per kilogram, but for vitamin E, milligrams per kilogram and international units are the same. But for vitamin A and vitamin D, you need to have a conversion factor. So what we're showing you here, even though maybe the papers gave us original data in international units, I've converted those into milligrams. So here we have fat-soluble vitamins requirements from another source. This is from a paper in a book, and the book is here, Novel Approaches Towards Sustainable Tilapia Aquaculture. It's I was unable to find it anywhere for you to be able to download it, but you can go and find it. Buy it if you want, but if you have, once again, an institutional subscription, you can get access to it and you can read the electronic version, or maybe you have a library that might even have a copy in the library. The reason that I included this table is now we see vitamin A and we see beta carotene. Now beta carotene is here because it's a provitamin A. Then we have vitamin D, vitamin E, vitamin K, and we see the recommended levels here are expressed in international units. But it does give us deficiency signs. So this is what happens when we do not have enough. And one of the problems when we talk about vitamin deficiencies is there are a lot of common similarities between all deficiencies. And you see the first one in every case is poor growth. And so it's extremely difficult to tell if you're just seeing poor growth. Sometimes you might not even assume that it's a vitamin deficiency, but it might well be. And so you do some blood testing. We can see here we've got low plasma hemoglobin, we've got low plasma prothrombin, and so on. But for the most part, we have things that we can look at, simple things like draw blood and test hematocrit and so on, to try to estimate what might be going on. Moving on to water-soluble vitamin requirements. So mostly here we're talking about all of this list in the vitamin B1, B2, niacin, biotin, folic acid, vitamin B6, pyridoxine, pantothenic acid, and vitamin B12. These are all B vitamins. And then at the bottom we have myo-inositol and we have choline. We have the species that was tested. We have the requirement that the papers were publishing. Now, when it comes to pyridoxine, they say, and that's this one right here, they say 28% protein is 15 milligrams per kilogram and 36% protein is 16.5. So what's the difference? Well, pyridoxine, vitamin B6, is involved in amino acid metabolism, and so the amount of pyridoxine in the diet can be related to the amount of total protein intake and the amount of conversion of amino acids and metabolism of amino acids that the liver is undertaking. We see the parameters here, and then you can see some of these papers also go back quite far. Now, in this paper, we're looking specifically at niacin. So the paper by Dr. Xiao from Taiwan, he is a very well-known researcher, nutritionist from Taiwan. He has done probably at least 15 to 20 papers on tilapia alone, on vitamins, on all of their basic nutrient requirements. He's really someone that you can look up in Google Scholar. or another place like that and find out all of the different papers that he has written. Okay, so this paper, utilization of carbohydrates in warm water fish with particular reference to tilapia. And it's a hybrid, so this is would be a red tilapia. And their conclusion is niacin is needed at 25 ppm or higher to promote utilization of carbohydrates. We see here what he has done is he has tested two types of carbohydrate. He's tested glucose, which of course is just about the purest form of carbohydrate. Um, um, a uh, Then he has dextrin, and dextrin is a polysaccharide, uh, not difficult to digest, but the body would process these slightly different. In the case of the dextrin, it would have to break it down into individual glucose molecules for actual processing in the liver through the citric acid cycle. But we see here we have the different levels of niacin 0, 10, 25, 40, 55, 80, 120, and 160. And then when we look at weight gain, feed efficiency ratio, we look at liver glycogen, and we look at hexokinase activity. So hexokinase is involved in carbohydrate metabolism. And we see that we have statistical differences between 0, and then we see some differences in terms of weight gain. For example, 25 is significantly better than 10, which is significantly better than 0. But when we go from 25 to 40 to 55, they are not statistically different than 25. So this, I believe, is one of the reasons why um, there is not uh, any need to go higher than 25 ppm. We look at FCR, we see the same thing. 25, 40, 50, 55 are not statistically different. Liver glycogen, 25, 40, 55 are not statistically significant. So the thing to learn from this paper is that in terms of vitamins, the amount of vitamin we need is not just simply, a single number picked up out of a book, but we do have to understand that the composition of our diets affects the amount of vitamins we might need, and we might have to consider supplementing at greater level because whatever we're doing is different than what the researcher who did the research gave us this number has been using. So we saw that for pyridoxine, higher protein in the diet suggested it should be a higher pyridoxine level. And here, higher niacin is suggested when we're talking about more carbohydrates in the diet. So that means that if we're talking about extruded feeds, floating feeds, 20 to 25% starch, or we're talking about maybe slinking or slow sinking feeds, which might have only 6, 8, 10% starch, then potentially we could look at that information and adjust our niacin levels maybe downwards from the levels that we find published. So here we have from the same book as I mentioned before, water-soluble vitamin requirements, and once again, we have different species of tilapia, and we also have the recommended levels and also the deficiency signs and the reference. Now, when I'm talking about water-soluble vitamins, I have not talked about vitamin C, so I've separated it out. So here we have hybrid red tilapia, blue tilapia, Nile tilapia, and gift tilapia. Now, one of the things that complicates vitamin C requirements is different researchers have tested different types of vitamin C. So the poorest one is L-ascorbic acid crystalline because this is sensitive to moisture levels, it's sensitive to heat, it's very rapidly oxidized. Whereas when we're looking at C2 monophosphate, C2 sulfate, or C2 polyphosphate. These are um, forms which are stabilized by the addition of a phosphate or a sulfate group on the 2-carbon, making them stable against heat and moisture, and so they're not broken. And so we see then the levels that are recommended, and the crystalline is 5 or 6 times higher than for the phosphate forms. And you can see here what they were measuring. So weight gain and maximum body storage, specific growth rate, and we have other parameters. And then the researchers are shown there as well. Now notice Dr. Xiao has done a lot of work with vitamins as well. And the last three, Solomon and Abdelghani, they are from Egypt. Okay, so macro mineral requirements. So macro minerals are minerals, calcium, phosphorus, magnesium, potassium, minerals which are normally put into the feed in percentage amounts, 0.5%, 1%, 2%. You see here the species and the parameter that was measured. They say freshwater calcium free. That means that the water had the calcium removed. You see here the requirements. Some of them are suggesting ranges. And we then have Nile tilapia, Mozambique tilapia down at the bottom, looking at magnesium. Interesting that Van der Velden says that for Mozambique tilapia that magnesium in fresh water was not required. I don't know about that. I'm sure that the fish needs magnesium. Now the question is, perhaps there was magnesium in the water that was being used to grow the tilapia, and I'd have to go back and look at that paper to find that out, because it doesn't make sense that they wouldn't benefit from having some kind of a dietary source unless they're in high magnesium water. Now, one of the things that people have discovered is that by adding some salt, some sodium chloride to feed for freshwater fish in the range of 1 to 2%, this improves the ion balance in acidic water and reduces energy required for homeostasis. Now recall in the second video that I was mentioning the fact that these fish have to adapt and tolerate different salinities, that will impact their ability to maintain their um, salinity, the osmolality of their blood plasma. And then we see that Nile tilapia fed 8% sodium chloride. That's actually quite a high amount. Adapted rapidly to brackish water conditions. So when we're talking about rapidly, meaning that maybe instead of taking 2 weeks to adapt, maybe they just adapted in a couple of 3 days. Go and read the paper and find out. Now, the thing with 8% sodium chloride is I would be very concerned about this diet if I was making it in a factory, that this would be a diet that might absorb moisture from the air very fast. This could be a diet that I would worry very much could become moldy extremely fast. Now, 8% sodium chloride is probably enough to prevent bacterial growth, but in terms of absorbing water out of the air, that would just automatically bring mold. So I don't know that I would ever really go that high. I would probably not want to go up there. I'd probably want to stay close, plus or minus around 2% maximum. Now, here we're talking about adding digestible calcium. So calcium ion 2 plus. And we might do that depending on if our water was soft water. So soft water has the hardness is low, the alkalinity is low, and then if we can do a simple water test. We might determine that our water is too soft and maybe adding some calcium to the diet might be a good idea. Phosphorus, on the other hand, intake is mainly from the diet because water in general, whether we're talking about fresh water or seawater, has quite low phosphorus content. And in one of the videos that I posted in sustainability, where I'm talking about water footprint, I actually talk about the chemical characteristics of water. Now, when we're looking at calcium and phosphorus, we have to look at them as a combination. And normally we have to have them in a fixed ratio between 1 to 2 and 2 to 1. The reason is that calcium and phosphorus and even magnesium are moved from the lumen of the gut into the body using non-specific protein carriers. And if we have too much calcium around and the carriers are just picking up the calcium, they will leave the phosphorus and the magnesium behind. And then we end up being deficient in phosphorus or magnesium. So in general, when we're formulating feeds, we want to have this calcium phosphorus ratio. And we can do certain things to try to interfere with this antagonism between these two. But nevertheless, uh, normally we rely on our feed ingredients. Maybe we might be adding extra phosphorus, some monocalcium phosphate, dicalcium phosphate to add more phosphorus. But even as we're adding MCP or DCP, we're adding calcium in as well. And then Lal from the University of Halifax in Nova Scotia, he then gives us this range of several species that calcium to phosphorus in the whole body, the ratio should be between 0.7 and 1.6. Okay, in terms of macro mineral requirements, what I have here is a photograph of a tilapia. The tilapia above has been fed sufficient digestible phosphorus. So you notice that the fish is kind of long, a very good natural body shape, whereas the tilapia below has a shortened backbone due to inadequate dietary phosphorus and possibly even zinc. This could be related to high phytic acid in the feed from using too much vegetable proteins and not supplementing enough additional dietary phosphorus. And below here we have two photographs showing you, this is from a project called the EU Fine Fish Project, where they were trying to understand why did we get fish body deformities when we switched to high vegetable protein diets. And so what they show you here is that they see there's a fusing of the vertebrae here, and you can see the vertebrae are fusing here. And it reduces in a fish that has the same number of vertebrae, but because the vertebrae are fused, the fish ends up being shorter. And from the perspective of Europeans, this is a deformity. It's an animal welfare issue. Okay, so micromineral requirements, otherwise known as trace minerals. So these are normally reported in milligrams or micrograms per kilogram of dry diet. And we have copper, zinc, and zinc. One in a, let's say the zinc first, the upper one is in a practical diet, and then the lower one is in a semi-purified diet. And they have the parameters that they measured: weight gain and bone zinc. And we can see that in the diets we have these different recommendations. Selenium 1 to 2, it's normally required in very, very low amounts. Actually, if selenium is too high, then it can actually become toxic. but it's at the core of all of these selenoproteins that are involved in antioxidant enzymes like glutathione peroxidase in the body. Okay, so this is a paper from Mass from 2020, Carbohydrate Utilization by Tilapia, a meta-analytical approach. So when we talk about a meta-analytical approach, it means that they have used the reported results from a number of different experiments, and then they've sorted through the results and they found studies that can be related to one another. And it looks like they have around 20 studies that they have used. And each one of these dots represents the results from a different study. So we see carbohydrate digestibility, and we see dietary starch level. So in the middle here, when we have 20 to 30% starch, which would be about equivalent of a floating feed for warm water fish like catfish or tilapia, we have digestibility ranging from a low of around 50% up to around 70, 80, 90%. That might be due to the way the feed is made. In general, when we use good preconditioning and we use extrusion, we end up at the upper end of this range. If we're just using pelleted feeds and not using the best preconditioning steps, we might be at the lower end of this range. However, when we go and look at the second one, so this is starch, and then we look at dietary crude fiber. So dietary crude fiber includes cellulose, lignin, so those are indigestible carbohydrates, and it might include some other non-starch polysaccharides. And so what we see is that the body doesn't have a whole lot of tolerance. Once we get up to crude fiber levels of around 8, maximum about 10% here, we see that they are very poorly digestible. Well, what's the issue with that? Well, the thing is that if you're talking about non-starch polysaccharides, but you're talking about cellulose and lignin, they basically fill up the gut, and then the fish just simply feels full and does not want to eat. They're not hungry. They don't have an appetite. But if we're talking about other forms of dietary fiber, so the non-starch polysaccharides like the glucans, mannans, xylose, xylans, and so on, they are a type of soluble polysaccharide, and they absorb water. So the problem with those is that they have a tendency to absorb um, water, and then they fill the gut with gel. And the high viscosity of the contents of the gut due to all of this soluble fiber then makes it very difficult for enzymes to work to find the substrates in the lumen, to digest the substrates, and then have the free nutrients being then getting across to the gut wall to be absorbed. So in general, we want to have low levels. With talking about something like shrimp, we typically want to have only around 2%, not more than 2.5%. With catfish, tilapia, maybe where maximum is going to be somewhere around 8. And it's only in fish like carps maybe that we would then want even potentially consider having fiber levels above 10. Now, I mentioned to you that when we look at this one and we see that there's quite a range here, that it might depend on whether the starch is pelleted or whether it's extruded. And so we see that over here, that when we're talking about pelleting, that the starch digestibility after cooking, the starch is not that well gelatinized, although there's not that much difference. This is still 91 or so, less than 91, and this is basically about 99. But extrusion always produces a higher digestible starch. And so sometimes we can get by with less starch in the diet because the starch is better gelatinized and more nutritionally available to provide energy. Okay, so here we have the same paper, and here they're talking about cellulase activity. So cellulase are enzymes that are active to break down any kind of fiber. And then in the case of tilapia, they don't have a lot of cellulase activity, but because they have a long gut with a lot of commensal gut bacteria, a very effective gut microbiota, in a lot of cases, those bacteria break down the carbohydrates. This is what we would call prebiotic fiber. So if we look at these two graphs, we have energy digestibility in percent against dietary non-starch polysaccharide level. We see that as the polysaccharide level goes up, that energy digestibility goes down. And previously I mentioned the fact that these different types of non-starch polysaccharides have different impacts. In this case, this reduction of energy digestibility, meaning that the material is not available, not able to be absorbed, is probably mostly due to the soluble um, non-starch polysaccharides being unable to be digested down into the basic components like uh, um, monosaccharides, disaccharides, or even down to glucose. Now, when the bacteria eat this, they will turn it into short-chain fatty acids, into acetate, propionic acid, butyric acid, and so on. And then when we look at feed conversion ratio against the dietary non-starch polysaccharides, we see that it goes the other way, that as we increase the NSP level, the feed conversion goes up, which is a bad thing. But notice that we have in here, we have such a wide range. We have from somewhere less than 1 up to more than 3 in terms of FCR. So these numbers are probably due to different ways that their feed was prepared. Although we have to go and read the paper and see the list of references to understand what exactly those diets that were being tested contained in terms of their non-starch polysaccharide content. Okay, so one thing that fish are kind of notorious for is for years and years and years, we've really wondered about the ability of fish insulin to control blood glucose levels, blood sugar levels. And so here I found this paper. It's actually quite difficult to find. So you have, it's coming from a paper published by one department of the Australian government. And there's a QR code. You can go and download it. And this is one of the papers in this report by Anderson. And what he's actually doing is he's comparing the glucose regulation of Asian sea bass juveniles. And then he has, just for reference, compared it to tilapia. He was actually more interested in Asian sea bass. But what we see is if we look at, okay, so what have we've got? We've got intraperitoneal injection of 1 gram, 2 grams, 4 grams of glucose per kilogram body weight. So that means that glucose was injected into the abdominal cavity at 1 gram, 2 grams, 4 grams per kilogram body weight. And what we see with the Asian sea bass on the left is glucose alone. is that as the level of glucose is increased, the Asian sea bass has a harder and harder time to reduce the glucose in the blood. Whereas the tilapia, doesn't matter whether you're giving it 1, 2, or 4, it is able to reduce the blood glucose very fast down to basically the foundational level. And when we look at more complicated carbohydrates, so we have galactose on the top here, and then we have xylose at the bottom, we see that once again the Asian sea bass is very slow. And look at the units we're talking about. We're talking about 30 hours, very long, 25 hours, quite a long time. And the levels are still very high. And if we're looking at this slope here, how long would it take to come back down to baseline? 2 days? 3 days? Very hard to say. Now, when it comes to xylose, the tilapia seems to have somewhat more of an issue. Maybe it doesn't have a lot of xylanase enzymes. And it would have to require, maybe rely on gut microbiota to get that job done. So this is just showing you that the absorption of glucose energy into the cells to drive the cellular energy cycles inside the cells is affected very much by the type of uh, starch or type of carbohydrate that the animal is fed, and the body's physiology and metabolism has a big impact on how it's able to use it and how long it takes to return everything to normal. Okay, so the last section, just a few slides here, dealing with red tilapia. I will actually have another video sometime about carotenoids, and I'll go into a lot more detail. But to just summarize, there are two classes of carotenoids. We have the first one, xanthophylls. So xanthophylls. So here we have canthaxanthin, astaxanthin, zeaxanthin. These are xanthophylls, and they contain oxygen atoms, and it makes them somewhat polar because they have a negatively charged oxygen molecule at either end. So here you can see there's an oxygen at this end and there's an oxygen at this end in both of these. But carotene, and the top one is a carotene, do not contain oxygen and are nonpolar. And so they behave much like fatty acids in membrane. So there's no oxygen at either end of this. Now, if you take this and you split it in half and it becomes 2 molecules of retinol, that then is essentially vitamin A. Now, the thing is that I have found that with tilapia, the xanthophylls seem to be somewhat better. But a lot of freshwater fish are very good at converting carotenoids into the highly pigmenting forms such as canthaxanthin and astaxanthin. I found a paper written a long time ago by a researcher, Harry Dupree at Arkansas, USA, who said that in order to minimize the yellow color in channel catfish meat, that you could not have a xanthophyll level higher than 11 ppm. If you had higher than 11 ppm, the flesh would be yellow, which was undesirable. So when I came to Thailand, then I said, well, it's obviously should be a lot higher than that. So then I determined that it needs to be at least 20 milligram of total xanthophylls We're not talking about carotenoids. Total xanthophylls or greater, and we can get that from our feed ingredients, like from our corn gluten meal, from our um, corn gluten feed, or from our DDGS, and so on. So we can obtain visually yellow-orange pigmentation in our fish with natural carotenoids. So we have beta-carotene, zeaxanthin, lutein, and cryptoxanthin. These can be obtained from ingredients such as alfalfa, corn gluten meal, and other corn products, as I mentioned. Now, when we feed zeaxanthin, like corn gluten meal, to um, red tilapia, they can develop a very nice red color. However, if we feed them something high in lutein, and one of the common things, for example, to color egg yolks is to add a supplement made from marigold flowers, which is very high lutein, this tends to give not into the flesh of tilapia, but into the skin, um, a very yellow tint, and it looks a little bit strange. But we can feed canthaxanthin, cryptoxanthin, capsanthin from paprika, and all of these will be converted into a nice color. Now, you may notice at the very beginning of my slide today, I showed you a picture of Nile tilapia that were receiving a diet high in xanthophylls, and you see a lot of red coloration on the fins, the dorsal fins, and the ventral fins, and then also on the tail. Now, the degree and quality of pigmentation from those ingredients is usually inconsistent due to production and processing conditions of those ingredients. And then in the feed manufacturing process, if we do not have enough antioxidants, or we have a diet mixture which has too many pro-oxidants, then those carotenoids can be destroyed due to our extreme processing conditions, high temperature, high moisture, and the oxidations occurring, and then we might have them in the feed, but the animal does not be properly pigmented. Now, we can also consider adding synthetic or supplemental pigments such as beta-carotene or apocarotenol, and we can bring out the color. And there's no need to use astaxanthin or canthaxanthin, because the animal will actually, if they want those, they can actually synthesize them from the more simple carotenoids. So here we see that this is just a sub picture from the one that I have at the beginning. You can see here these black tilapia have pectoral fins that are orange, and the caudal fins are also orange. And then hybrid red tilapia can utilize xanthophylls, and it colors the skin, but it does not pigment the flesh. Whereas if we're talking about catfish like channel catfish or uh, clarius catfish, not only does the skin get pigmented, but also the flesh will become yellow. So I suggest supplementing to reach 40 ppm total xanthophylls, especially if we're not being really careful about our antioxidants and our pro-oxidants. The minimum xanthophyll level should be 25 ppm, and that I would try to avoid having ingredients high in lutein if at all possible. And this fish that you see here on the side, it doesn't show up very well, but if you look in this area, you see it's white. But if you look up in this area up in here, you see that there's actually a lot of yellow there, and it doesn't look very natural, although it's probably not going to affect anything about the quality of the fish. Okay, so as I finish up here, let's talk about some resources. Let's talk about some places that you can find information that will allow you to understand more about nutrition of tilapia. So this is a paper that I highly recommend, a review of the nutrition and feeding management of farm tilapia throughout the culture cycle by Dr. Wing Kyung Ng in Malaysia. There's the barcode. It is not available free, but it's a very long review, and it has lots and lots of tables for proteins, amino acids, lipids, fatty acids, carbohydrates, vitamins and minerals, everything. And I just show you one example. You can't really read it here, but that's not the point. The point is you see the tables. This is just one, are very detailed, and if you can have access to this paper, I really recommend that you find it. These are a number of other things that I recommend. We have over here 2 books. Um, they're not available to download as far as I can find, but both of them are available on the Internet Archive, and you can read them online. And then, of course, you could probably also find something you want and screenshot it, but that's up to you. This one I recommend because of this chapter written by Professor Johnsey from Sterling University on nutritional requirements of tilapia. So that's very good. And then Chorn Lim is one of the main researchers for catfish and tilapia in the United States. So we have various other papers here. Use of ideal protein concept for precision formulation of amino acid levels in fish meal free feeds for juvenile tilapia. And then we have El Sayed. I mentioned him before. He's the one who's written numerous papers and books about tilapia nutrition. We have here Dr. Xiao from Taiwan writing about vitamin requirements. We have these papers, interactive effects of water, temperature, and dietary protein on growth, immunity, and physiological health. Use of ideal protein concept for precision formulation of amino acid levels. Ideal protein concept is something that is being widely used in poultry and also in swine. It has been tested with various fish species, and it seems to be a fairly decent way to potentially set your amino acid levels in your feeds. We then have several papers here on aquaculture nutrition, and by researchers who have written quite a few papers about nutrition in fish, not necessarily all about tilapia. This paper here, contributions of direct incorporation from diet and microbial amino acids to protein synthesis. I will come back to that later when I start talking about the gut microbiome, because this is talking about the fact that when the fish obtains its amino acids, is it obtaining them from the diet, or is it obtaining them because the gut microbiota are synthesizing them? So they have some interesting techniques that they use, including tracking carbon isotopes to see what's happening there. But it's a lot of detail, and it's too much to talk about here. So finally, we're done. And this is a picture of a layer chicken open barn over a tilapia pond, a very large tilapia pond in Chiang Rai, Thailand. So please subscribe to my YouTube channel, and then like, make comments. If you ask for something, I may be able to find something else to do for you. Turn on notifications so you receive more information about when I post new videos. And particularly if you're a student, either at a high school or university level, please share my content with your friends, and so that everybody can learn more about fish nutrition.